So good morning everyone and welcome to the session. Welcome to the next session of the series of immunology and immunotechnology. And today we'll be talking about T cell receptor signaling. As I told you clearly that today we'll be talking about from where we left in the past. Now where we left in the past, we had a very good narration and discussion regarding your T cell immune responses, that how a T cell is mounting an immune response, how it is getting activated, and what are the effector functions of after getting activated by that particular T cell. In today's lecture, what we'll be discussing about that if this is a T cell, if this is a T cell and this is its receptor, now receptor will not look like that, but if this is a receptor and it is getting activated with an antigen presented with an MSC molecule, then what is happening inside this T cell? What is happening inside this T cell that is eventually leading to the activation of immunoregulatory transcription factors and release of cytokines like IL-2, 4, 6, 10, 5. So all those repertoire of cytokines that are required for T cell mediated immune responses, what is exactly happening inside this T cell, and what is the role of this particular receptor in so-called signal conduction or signal transduction inside that particular cell. So that will be our main objective, is just to understand what are different types of T cell receptors that are present inside our body, and based on those T cell receptors, how we categorize them into different categories. All the categories we'll be looking at, and then we'll be talking about a very brief understanding based on animation also. So let's start. Let's not wait further. Let's directly dive into the topic. And today as I told you, that we are talking about a T cell receptor. So let's talk about this image. Let's not worry about the text what we have written. Let us first focus on this image. Now, this is your T cell. I'm not talking about what kind of T cell it's there. This is your T cell currently. And then this T cell is getting activated with the APCs. Now, this is nothing new. This we have already established. Fine. This we have already established, and this is what is leading to activation of T cell. Now, this is these APCs are what? These APCs are antigen presenting cells like macrophages, B cells, and dendritic cells. Okay. So this macrophages, B cells, and dendritic cells, they express the processed antigen in the form of peptide bound to MHC molecule, and that complex in this complex is recognized by T cell receptor. Now, our job today is to understand what is this T cell receptor. Now, you should first understand that this T cell receptor, as evident in the picture, I have represented it with two colors. Now, there should be a reason that I have represented it with two colors, simply because it is made up of two different polypeptide chain. This one is alpha chain, as we call, and this is beta chain. Then there will be another type we will be calling gamma and delta. So based on the expression of a polypeptide chain on the surface of T cell, we categorize T cells based on T cell receptors. Now, when we are talking about the T cell receptor, which is making close contact with the MSC molecule or the peptide antigen, these two membranes are in very close vicinity to each other. And that is a reason why T cell activation and T cell recognizing the MSC molecule is very complex, what we have discussed compared to your T cell activation. And just to start with the thing, I have incorporated a video which is a very beautiful animation. Let's look at this and try to have a very basic understanding of how the things are happening inside that particular aspects of T cell receptor. Now, when we are talking about T cell receptor, see T cell, again, there is nothing new that I'll be talking about in terms of initial understanding that T cell is, we are talking about T lymphocytes here, and T lymphocytes are produced in bone marrow, and from bone marrow, they are, after being produced, they are reaching to a thymus where the maturation is actually happening. So that is a basic understanding that you should always have in the back of the mind. So when we are talking about the maturation in the thymus, the maturation is coming in aspect in terms of your receptors that are expressed on the surface. And if you see, then again, we have expressed two straight lines in form of receptors. And these two are nothing but the chains that I discussed in the previous slides, alpha and beta chain. Now, a single T cell receptor after getting mature contains thousands of copies of these T cell receptor on the surface. So we, for the simplicity sake, I have showing only two copies, but there are thousands of copies that are present on the complete T cell membrane. Now, these T cells will be expressed, as I told you, after getting exposed to the antigen only in the thymus, and their expression is what we are calling in terms of maturation part. So, when the T cell receptor is being formed, then what happens? It recognizes the MSC molecule which is presenting the antigen, You see, the red color antigen is expressed is presented by the MSC molecule of antigen presenting cells. Now, antigen presenting cells may be dendritic cells, macrophages, or B cells, depending upon what type of immune response are we talking about. So, we have more than 10,000 to 3,000 identical copies of T cell receptor, and based on the surface expression of the T cell receptor, we categorize them into two classes: alpha beta and gamma delta. Alpha beta is almost 95% of the T cells that we will be seeing inside our blood, whereas on gamma delta only 5%. Now, why 5% and why this 5% is important, that we'll be talking about in the subsequent slides. But you have to understand that what is the basic difference between these two. What is the basic difference between these two? So when we are talking about T cell receptor alpha and beta, it is composed of two polypeptide chain. This is the membrane. It contains an alpha chain and it contains a beta chain. Now, if you take out this receptor, from the cell and you go for electrophoresis of SDS page, you will clearly see two different protein bands. That means a band of 43 kilodalton and another band of 49 kilodalton, representing alpha chain and beta chain. So that is the reason why we are talking about that T cell receptor is made up of alpha and beta chains. Now, when we are talking about alpha and beta, we call them as heterodimer. So T cell receptor is always a heterodimer. In the alpha chain, we can divide it into two parts. The upper part is variable region, which is responsible for binding to the antigen, which is presented by the MSC molecule. Another one is constant region, and both the regions are bounded by disulfide bonds. So the highlighted portion yellow is nothing but an antigen binding site, which will be responsible to bind to the processed antigen, which is presented by the MSC molecule. It will never bind to the soluble antigen. The antigen which is floating in the blood, which is not processed by the antigen presenting cell, it can never bind to that particular antigen. It will only bind to the antigen which is presented by MSC molecule. Now, next phase that is very important, let me tell you before going ahead. Just give me a second. See this picture. If you see this particular portion which I am highlighting, the region which is spanning the membrane, the region which is penetrating the membrane. Now, this particular region which is actually penetrating the membrane, you see at the end there is a very short length that is actually entering inside the cytoplasm. So consider this is a membrane, this is a cytoplasmic material. A receptor, ideally a receptor should be, in this scenario, that it should be able to transduce the signal via cytoplasmic tail. The cytoplasmic tail, the longer the tail, the better transduction will be there. So this currently alpha beta is having a very short fragment. In fact, I can tell you it's only 3 amino acids that are present inside the cytoplasmic part. So we understand that it will not be able to transfer the signal that is given by after antigen binding to the cytoplasmic domain. That is the reason why a T cell receptor requires an additional co-receptor in the form of CD3 molecule in order to give the signal, to cytoplasmic domain, and that we'll be talking about in subsequent slides. So let's go ahead from where we left. So there's a T cell receptor which has recognized the processed antigen presented by the MSC molecule, and then after attaining the exposure, they will start signaling. So the T cell receptor cytoplasmic tail is very short. It contains only 3 amino acids. Now these 3 amino acids are not enough to transfer the signal to the nucleus. It's as simple. You see, in the previous understanding of B cell activation, we had established an understanding that there are motifs called as items. item notice should be phosphorylated, and then the adapter proteins will be coming, and then the phospholipase C will be activated, and then so on. Similar scenario will be happening in this particular signal transduction. So, in order to transfer the signal to the nucleus, the T cell receptor is associated with another set of proteins that are called as co-receptor, and collectively those four different proteins are called as CD3, cluster of differentiation 3, which is always present on every kind of T cell. So what is the composition of CD3? It is a heterodimer. Now let us see what's a heterodimer. It's a heterodimer of two different types of chain. One will be delta and epsilon, another will be gamma and epsilon. Now many people ask me before starting, what is this alpha, beta, delta, epsilon, gamma? Why the nomenclature is there? Now, after understanding this particular aspect, when we going for genetic level, you will see that the gene which we'll be talking about contains the regions that are specified for this particular polypeptide chain. And just to have a nomenclature in the immunoglobulin family, the complete nomenclature is provided in the form of alpha, beta, gamma, delta, and epsilon. To differentiate, you just have to remember that there are 5 different types of chains that we'll be talking about, and we'll be talking about alpha chains, beta chains, gamma chain, delta chain, and epsilon chain. So there are 5 different chains. Of course, for those 5 different chains, there will be 5 different segments inside the chromosomes that will be coding for this particular polypeptide. Then, if we are talking about the CD3 part, then CD3 part specifically is made up of 2 separate heterodimers. One is in front of you, that is gamma and epsilon. Another one is delta and epsilon. And not only this, sometimes along with this, there is an additional homodimer of zeta chain that is also present. So if somebody asks you that on the T cell, what is the composition of T cell receptor? It's very simple. T cell receptor is alpha and beta chain. That is here. It is a heterodimer made up of alpha and beta chain. But unfortunately, because of the length of the cytoplasmic part that is very short, it is not able to transfer the signal to the nucleus. So it requires the help. It requires the help of the other receptor molecule which is present in the immediate vicinity adjacent to this particular T cell receptor. And that molecule is called as CD3. Now CD3 is present in all the T cell types, that and delta epsilon. Now, if you see the structure here, it contains only one loop, and this loop is called as immunoglobulin superfamily identification mark. Any polypeptide which forms a loop like this is categorized under the family of immunoglobulin superfamily. So this contains only one loop, and it's a constant region. So you can see there is no variable region. And that is the reason why this particular CD3 cannot bind to any antigen. That antigen binding is the characteristic property of variable region, which is present only in the T cell receptor. So the job of the variable region is to bind to the antigen and then ask the CD3 molecule to help in transducing the signal. And it helps because of these yellow regions that I have highlighted in the cytoplasmic tail. Then these yellow regions are nothing but your item molecules that we had discussed in the past lectures. Then I told you that there is another set of chain, that is zeta chain, and you can see that there are multiple item molecules that are present. So if suppose these 3 molecules are collectively at CD3 are helping this, then the transduction or conveying the message becomes very, very easy for the cell. Very, very easy for the cell. Now, if we talk about different types of T cells that are present, this zeta chain and the CD3 proteins are identical in all the types. The difference in the T cells are arising because of the T cell receptor. Because of the T cell receptor, and experimentally you can easily identify these particular things. You isolate T helper cell, you isolate cytotoxic T cell, and you isolate gamma delta T cells, and you check all the receptor morphology, receptor components. You will see that these 3 proteins, alpha, beta, delta, and epsilon, zeta are delta, epsilon, and gamma, zeta. These 4 are constantly present, conservatively present in all the types of T cells. The difference is only on the basis of T cell receptor, only on the basis of T cell receptor. And this complete part cumulatively is called as T cell receptor complex that forms a complete motion. This is a T cell receptor, and the complete complex is made up of CD3 molecule and a pair of zeta 3 chain. And that is the part that we wanted to have a very clear understanding regarding that scenario. Then let's go based on after attaining an insight based on the animation part. Let us directly go and discuss how these things are happening. So we discussed that there are two types of T cell receptors. What types of T cell receptor I'm talking about? I'm talking about the chains of alpha beta. This is making up one type of one class of T cell that is called as alpha beta T cells, and they constitute almost about 95% that we have established in terms of the understanding of animation. And separate set we have gamma delta T cells, and you see these days gamma delta T cells, or not these days, from past decade or 2 decades, these gamma delta T cells are regularly being explored. That why this 5% of the population that is there in the T cell, how they are originating, what are the root causes that they are originating. And during the lecture, in this lecture, you will see that if this particular alpha beta is only specific for the processed antigen presented by the MSC molecule, this barrier is not there in case of gamma delta T cell. It can go and bind to the glycoprotein. It can go and reside in the mucosal tissue and perform different functions. So there are many functions that are performed by gamma delta T cells, and utilizing their properties were the prime target in clinical settings. So let us talk about a certain aspect that because of the presence of gamma delta chain, we are calling these 5% of the population as gamma delta T cells. But both the type of the cells will be having CD3 molecule, and CD3 I mean the two heterodimers and the homodimer of zeta. They will be having a very short cytoplasmic tail, that's common part. They will be having a transmembrane region, a single constant region, and a single variable region. So if you compare this receptor with the B cell receptor, what was the B cell receptor? B cell receptor was membrane bound immunoglobulin M. It contained two sites of your antigen binding, whereas here we are having only a single site. That is the reason why we call it as monovalent, and we call the receptor of B cell as bivalent. Now, when we are talking about the second subset of T cell that is expressing gamma delta T cell, it is residing in the majorly in the mucosal tissue, and it contains a specific pattern that is associated with the gamma delta T cells, which can be easily recognized. This part. Now, these gamma delta T cells can bind to the lipid molecules, they can bind to the glycolipid, whereas I told you that alpha beta is specific for the processed antigen, and then processed antigen is nothing but a peptide. So the barrier of binding to a peptide is crossed by the gamma delta T cell. Now they can recognize the lipid part also. So we can use these cells to target certain viral proteins, certain bacterial membrane targets that are expressing a unique pattern of glycolipid. If we have an immune cell that can target the lipid portion, so these gamma delta T cells are the candidates that are targeting lipid and glycolipid part. And not only this, people have shown that these gamma delta T cells also target certain heat shock proteins. These heat shock proteins are our own proteins, so they have the potential to target the endogenous antigens also, or in terms of phosphoantigens, which are phosphorylated, that are derived from microbes. So the spectrum or the diversity in terms of recognition of any antigen is very vast in case of your gamma delta T cells. So one should have a very clear cut understanding that if we are talking about alpha beta, I'm talking about the peptide presented by MSC, and if I'm talking about the gamma delta, I'm talking about different targets, the lipids, the glycolipids that are of course of foreign origin. Then we have phosphoantigen derived from the microbe. But if you're talking about our own generated protein, then the heat shock proteins, a certain heat shock proteins are recognized by gamma delta T cells. And people have exclusively shown, after isolating these receptors and going for crystallization and comparing the X-ray crystallography structure with the alpha beta TCR receptor, they clearly established that the domains the variable and constant region that are showcased here, the variable and the constant region, the actual alignment of these chains, there is a difference between the bond angle, and that angle difference might be responsible for this much diversity when we are talking about in terms of gamma delta T cells. And that is a reason that that people across the scientific community are projecting that the difference between this bond angle might be the case. This angle I'm talking about, this angle, the difference between the variable and the constant region. We have represented how we have represented. We always show in picture that's a straight line, but in terms, it's not a straight line. It is actually a bended part that you will observe in case of T cell receptor. So once you have understood the complete etiology of the T cell signaling or the T cell receptor, then how that signal is transduced inside the nucleus, we understood that there is a complex called as CD3. Now this CD3, I told you that it's a heterodimer of gamma delta or delta epsilon. It's made up of gamma epsilon or delta epsilon. So there are two heterodimers, and then occasionally there is a homodimer that is present called as zeta chain. Now they have a huge length of cytoplasmic domain. Now this cytoplasmic domain is nothing but the item molecule that we are talking about, and this particular item molecule will serve as a site for phosphorylation and will serve as a site for binding to the proteins which contain the SH2 domain, as we discussed in the previous lecture. So this CD3 complex is made up of these molecules, and this is actually responsible for transducing the signal. And please remember, collectively CD3, homodimer of zeta chain, and T cell receptor. Collectively, these three are pronounced or called as T cell receptor complex. T cell receptor complex that we are talking about. So that is the case. And now, as I told you, that what is the function of these CD3? That's the next question. Now, I told you that the cytoplasmic part is nothing but the item molecule that we are calling as immunoreceptor tyrosine based activation motif. We had a very clear understanding that what is that motif in the previous lectures. Now, let's recapitulate what we meant by that particular target, what we meant by that particular item. Now, just to give you an example, say this is the membrane, and this is the cytoplasmic domain, and I have a item region. Now, item region, the M stands for motif. It's a very short region in the polypeptide chain, which is doing a characteristic function. Now, this item molecule, upon binding with the antigen, will be getting phosphorylated. Who will phosphorylate? We will see. It will get phosphorylated. Now, the phosphorylated item motif serves as an adapter site for the proteins that contain a specific domain, and that domain is called as SH2 domain that we had discussed in the earlier lectures. This domain, which will be binding to the phosphorylated site, this domain is called as SH2 domain. What do you mean by domain? It's the small region of a three-dimensional structure protein which can perform a specific function. So what is the function of SH2? All the proteins which contain this particular SH2 domain can bind to the phosphorylated protein, and that is the exact function of SH2. So they can go for a docking kind of scenario in terms of attachment. So when we are talking about the CD3 dimers, these dimers, why they are not combining, why they are not associating with the T cell receptor? You see, in every pictorial representation, I am giving you a certain distance that is there between these two molecules. Now, why they are not directly attaching? Simply because the amino acid residues that we are talking about in terms of their constant region, they are oppositely charged. Now, how? See, the T cell receptor are positively charged, whereas the CD3 molecules are negatively charged. So there is the association or the ionic interaction that is there always to hold the complex together. It will not let the CD3 molecule sway away in the membrane part. Because of these amino acids, because of the charge that is positive here and negative here, there is a kind of attractive force that is being applied by T cell receptor to the CD3 molecule, so that this particular entity should not go away from this T cell receptor. It should be maintained as such as T cell receptor complex, and that maintenance is because of the ionic interactions of the amino acids. So in real time scenario, if you observe, if you see that how the complex is showcased, then this is the real time picture. This is the real time picture that the T cell receptor contains 5 disulfide bonds. Again, a very important aspect in terms of your questions that are asked in competitions. How many disulfide bonds are present in antibody? More than 14. How many disulfide bonds are present in T cell receptor? 5. How? You can see here one which is holding up the domains. Now we have 4 loops. The 4 disulfide bonds are actually responsible for looping the complete chain, and then both the chains are associated by a disulfide bond so that they are static. And the transmembrane part, the transmembrane domain contains positively charged amino acids. Then we have CD3 molecules. CD3 molecule contains each heterodimer contains two disulfide bonds, and negatively charged amino acids in the transmembrane domain. Now that is the reason why these negatively charged domains are in close proximity to this particular T cell receptor. Then they contains multiple item motif. Now, please remember the CD3 molecule, if you are talking about the heterodimer, contains 2. Each chain contains a single item motif in case of heterodimer, whereas the homodimer of zeta contains 3 item motifs in each chain. And these blue color regions are actually responsible for transferring the signal to the cytoplasm and from cytoplasm to nucleus. See why the signal, why after binding an antigen, a signal should be delivered to the nucleus. It's very simple, because in nucleus the DNA is present, and once the activation signal is given, then and then only the inflammatory proteins, the mediators, the interleukins, the cytokines that are required to give the signal to the other cells will be getting released. Fine. So that is why the complete activation part has to be there. That is why the complete activation part has to be there. Now, let's summarize. Before going ahead, let's summarize how the signal transduction is happening. What are the accessory molecules on the T cell receptor that that plays their role, specific roles in T cell signal transduction? We understood that yes, there are different molecules. We have a T cell receptor that is bind with disulfide bond. We have CD3 that is there, a complete complex. What apart from this? See, we have a CD4 molecule that will be binding to class 2. We have CD8 that will be binding to class 1. This is in T helper cells. This is in cytotoxic T cells. Apart from this, we have a very effective co-stimulatory pathway that is CD28 and CD8086. Now, what is this CD8086 and CD24 that we'll be discussing in the subsequent slides? See, we have first co-stimulatory signal with CD28, CD8086, then we have CTLA4, then we have CD45R. These 3 are very important in terms of T cell activation, and how these 3 actually contribute to T cell activation, that we'll be seeing in the coming slides. Now, when we are talking about the core receptors, that is CD4, CD8, we already have a very clear-cut understanding that if we are talking about T helper cells based response, it is CD4. If you are talking about the cell-mediated immune response, cytotoxic T cell activation, we are talking about in terms of CD8 molecule. Then how these necessary molecules are helping in transferring the signal. Let us understand. See, this is the MSC molecule which has presented the antigen, and on every cell, the alpha beta chain that is there will be responsible for binding to this particular processed antigen. So your T cell receptor is actually binding to the MSC class 1 presented antigen. But in CD8, positive T cells, the CD8 molecule will go and bind to the MSC part and will bind to the MSC class 1. Thereby, it will recognize that yes, now the signal has to transfer. So this is the first co-receptor signal that is generated in terms of your T cell receptor signaling. That once the T cell receptor has recognized the antigen, then CD8 molecule will be binding to the MSC1, and if it's MSC2, then CD4 molecule will be going and binding to the MSC class 2. After these bindings have been attained, then the next job is to call the CD3 and let's start communicating what we have attained the information to the nucleus. Let's start communicating the information to the nucleus. That is the next job. So let's have a brief glimpse of what is CD4 and CD8. Let's have a comparative chart. This is CD8 that I am talking about, and the left hand panel that we are discussing is actually CD4. So this left hand panel, what we're talking about, CD4. CD4 is of 55 kilodalton. CD8 is made up of 30 to 38 kilodalton, 30 to 38 kilodalton glycoprotein. It's a very small glycoprotein. It is made up of you can see in the clearly in the picture. It is made up of two different chains. It is made up of two different chains, right? So that is the reason why I'm calling it as a homodimer. It can be a homodimer of alpha alpha or beta beta, and it can also be a heterodimer of alpha beta. There is no notation. In both the cases, you will see that the two chains are falling under the category of 30 and 38 kilodalton. Whereas when we are talking about CD4, it contains a single chain. It contains a single chain, a membrane glycoprotein in the form of monomer with 55 kilodalton protein. So, what is your job? Let me give you a very simple job. Design an experiment to showcase or to prove that the CD4 and CD8 molecules that are present on T helper and cytotoxic T cells are of 55 kilodalton and 30 to 38 kilodalton proteins. It's a very simple experimental setting. Once you have a very clear understanding of what we have narrated in this particular lecture, perhaps you can go about and design that experiment. And if you are not able to design, you can just attend the forthcoming lectures to have an understanding that how these statements are given. See, a statement is very simple. CD4 is 55 kilodalton, but in order to determine this particular statement and put this particular statement to the scientific community, the experimental settings are required, and that experimental setting is the job of us to understand clearly. Now, this particular 55 kilodalton contains four domains: D1, D2, D3, D4, and similarly, there are multiple domains that are present in CD8 also. So this is the representation. It contains 1, 2, 3, 4 domains, whereas in CD3 it contains only 2 domains and a long stalk region that is there in order to when we are talking about the CD4 and CD8 aspect. So this co-engagement that we are talking about along with the MSC, the co-engagement of CD4, CD8 with the T cell receptor, this actually enhances the capability of T cell to bind to their target. If you remove this CD4, CD8, the affinity of T cell receptor to bind to the antigen presented by the MSC is exponentially reduced. So that is the narration that we wanted to have completely. We wanted to have in this particular session, that you should be able to understand first that how the things are happening, what is T cell receptor. Then in subsequent lectures, we will be talking about how exactly the signal is generated and transduced to the nucleus, and then how these T cell receptors are actually formed. So till then, I request all to please go through the lecture and understand that how these things are happening and how these particular aspects are coming together to give rise to a T cell receptor mediated response. So just to brush up, these are the MCQs that you should focus on, and perhaps these MCQs will will promote your interest and will create a curiosity, ignite a curiosity in terms of understanding the topic. So thank you so much. And this is all that we wanted to narrate in this particular session. And we'll again meet in another session talking about the T cell receptor. Till then, keep learning and wish you a very happy and safe learning scenario.